Hello, this is Eva Amson, welcoming you to this Bite-sized Bio webinar, which today is sponsored by Twist Bioscience. Today's presentation is titled Using Plant Synthetic Biology for Sustainable High-Yield Rubber Production, and it's being presented by Julia Rin and Alexander Kuhne. Julia and Alexander are grand prize winners of the 2024 iGEM competition, specializing in molecular and cellular biology at Philips University Marburg. In today's presentation, you will learn more about their award-winning work. As always, we will have a question and answer session after the presentation. So please type any questions you have into the questions box, which appears at the top right of your screen, and I'll put them to Alexander and Julia at the end. Details of how to access the on-demand recording of this webinar will be sent to you by email shortly. So now over to you, Julia, for the presentation.
All right, thank you very much. Thank you so much for having us. We're very excited to talk about our iGEM project 2024, where we looked into dandelion engineering and its potential for sustainable rubber production. But before we jump into our project, we would like to talk about a bit about iGEM and tell you something about that. So iGEM is an international competition for synthetic biology where student teams from all over the world work on a real-life project using synthetic biology. And these projects can be very, very diverse. So you have teams that work on projects in agriculture, others work in diagnosis or foundational advancements. There are even teams that work on projects related to space. So in the end, all of these teams come together at the big finale in Paris, where they showcase their projects, they're being judged by a jury, and in the end, the grand prize winner is announced. And last year in 2024, that was us with our team, the iGEM team Marburg, and we won with our project Taraxacum, and this is what we want to talk with you today about. Now, as every good story, we would like to start in the beginning. Now, when we started with our project, we originally were interested in finding alternative bacteria for plant transformation. And for that, we were looking for model plants, but we did not want to use the usual ones that you have, like Arabidopsis, for example. We wanted to use other ones. And while we were searching for that, we stumbled upon a special dandelion species from Kazakhstan, which is Taraxacum kok-saghyz, a dandelion species that can produce natural rubber in its roots. And you can see that in the picture in the top right, these white strings that you see there, this is natural rubber. And this is very interesting because we all need natural rubber. We need it for over 50,000 products. So from simple rubber bands to shoe soles, to tires, medical products, natural rubber can be found in so many products. And for that, we require over 15 million tons of natural rubber each and every year. And the demand is rising. But we need this natural rubber. We cannot produce it synthetically because it has very unique properties that we cannot copy synthetically. So we need plants to produce that for us. In general, there are over 200 different plant species that produce natural rubber as part of latex. Now, the plants use it as a defense mechanism. They use it to seal wounds or to use it against insects that try to eat them. So when they bite into the plant, they get into contact with the latex, it sticks to their mouth, they cannot hurt the plant anymore, and they also, as a result, starve to death. But we for sure cannot use just any natural rubber for our industry. It needs to fulfill certain characteristics. So one is it has to have a high molecular weight to ensure the strength and elasticity of the material. Then we have to have consistent quality to fulfill manufacturing standards. And we for sure also need a sustainable yield so we can meet the industrial demands. Now, with these characteristics in mind, the 200 plant species that produce natural rubber are boiled down to 3. And the first one is the guayuli, which is a shrub that grows in rather desert areas. Then we have our dandelion species that grows in European latitudes. And we have Hevea brasiliensis, or as most of people know it as the rubber tree. And this grows or is cultivated in Southeast Asia mostly. And 90% of our natural rubber supply is harvested from the rubber tree. So we more or less rely on just this one plant species for this very important natural resource. And that comes with many problems. So since 1993, more than 4 million hectares of rainforest have been destroyed due to rubber cultivation. That's an area comparable to the size of the Netherlands. And furthermore, the trees are highly vulnerable to diseases, and issues like labor exploitation and abuse persist in the industry. So when we found that out during our research, we thought, okay, we need to have a diversification for our rubber sources. And that's where the dandelion comes back into place, because there are many advantages that it has. For example, it can be grown in European latitudes, so you do not have to cultivate it in tropical zones. Then it can be grown on low nutrient soil, so it does not necessarily have to compete with the food production. The rubber has a very high quality, comparable to the rubber from the rubber tree, or sometimes or even better. The chains are about 50% longer of the polyisoprene chains. It can be grown annually, so it can supplement crop rotation, and it can also increase biodiversity as a potential food source for insects. And at this point, you might be wondering, just as we did, there are so many advantages. If the dandelion is so great, why is it not being cultivated on a large scale yet? Why don't we use it as an industrial crop? And for that, I would like to compare the dandelion to the rubber tree. Now, if we imagine we have 1 hectare of land, we can either plant 2.5 to 5 million dandelions or 650 rubber trees on that 1 hectare. After 1 year, we would get from our dandelion field up to 1,000 kilograms of natural rubber. But that's not a consistent number. It could also just be 400 kilograms. From the rubber trees, on the other hand, we would get 1,500 kilograms consistently of natural rubber. And at that point, we thought, okay, what can we do about that? Can we maybe just increase the rubber yield in the dandelion, make that consistent, and then it's more or less the same as the rubber tree? But actually, although that's maybe the closest you can think of, that is not the solution, because the rubber yield in the dandelion is already at its maximum. So through crossbreeding, it was possible to get it up to 19%, and that's the maximum. If you go higher, the plant will die. So that's not the issue. The actual issue are agricultural traits, such as the seed size, root architecture, and leaf morphology that hinder the dandelion to be cultivated on a large scale. So we then focused not on the rubber yield, but we switched it to focusing on supporting mechanized harvesting, so that you have a standardized plant, and that farmers, for example, can use the machines that they already have. They do not have to invest in new machines, or that new machines have to be bought. But to change these characteristics of a plant, you need to have a genetic access. And this genetic access was not given when we started our project.
Yeah, thank you, Julia, because this genetic access is something we have worked on last year. So if you want to work on a genetic access, you obviously need to characterize the genetic parts you are building. And for that, you need rapid plant testing systems. And herefore, we focus on 2 different approaches. First, agrobacterium-mediated leaf infiltration and protoplast transformation. In agrobacterium leaf infiltration, you take an agrobacterium solution, so agrobacterium is a bacteria, and you basically just squeeze it into the leaf material of a plant, and the agrobacteria will transform the plant. And to show that our transformation is actually functional, we worked with the Ruby reporter in Dandelion, which is an expression system which produces over an enzyme cascade red pigment, which you can see on the right-hand side in the pictures over there. However, obviously, if a plant might be stressed, they might also have some different coloring, also some different red coloring. And to show that this pigment is actually the pigment we want, so betalain, we did a betalain extraction and also showed that this is actually the pigment we are looking for, so betalain, and our transformation is functional, therefore showing that leaf infiltration can be used for our approach. Secondly, we used also protoplast transformation, which is more of a high throughput approach because you can easier transform a very large amount of plant material this way. And here we showed that this works using fluorescence reporters. So you can see we were successful in expressing the M-cherry fluorophore and EGFP as a fluorophore in protoplasts at the same time after investigating them via fluorescence microscopy. Now, where we have shown that we are actually able to transform the dandelion plant in general, now we obviously need also the genetic material we actually want to test. And as Julia already has mentioned, excuse me, previously the genetic access for dandelion was just not there. Therefore, we looked into publicly available RNA sequencing data and built our own transcriptomic pipeline around it, making sure to filter for high expression strength and low variance, ensuring that our genetic parts are most likely constitutively expressed in the dandelion plant. Also, we ensured that our parts are phytobrick compatible, so most researchers have access to these parts and can actually use them easily in their approaches. Now, where we know where the parts are looking for, obviously we are not interested in the coding sequences, but in the regulatory elements of the promoters and the 3' UTRs, which we identified for the promoter 5' UTRs by taking a region 2 kilobases in length upstream of the coding sequences, which ensures that the core promoter elements and cis-regulatory sequences are included in this element, making sure it should work in other areas as well. On the other side, we used the 3' UTRs I already have mentioned, and we identified them based on previous genome annotation, but included 150 base pairs additionally downstream. This ensures that other regulatory elements, such as far upstream elements and cleavage sites, are also included in this 3' UTR, making sure this element should also work later on. Now where we know which genetic material we are working with, we also need a cassette to measure and to characterize these parts. Herefore, we used a ratiometric approach. Thereby, this approach works by using two different cassettes, the normalization cassette and the measurement cassette, which are harbored on the same plasmid. The M-cherry reporter is thereby controlled by the ubiquitin promoter 5' UTR and the NOS 3' UTR, and will stay the same for all of experiments. On the other hand, we have the measurement cassette with an EGFP variant. This is normally controlled with a 35S promoter and a 35S 3' UTR. If we now want to test a promoter from our toolbox, then we will just switch out the promoter of this cassette, and if we want to test a 3' UTR, we will switch out the 3' UTR of this cassette. Obviously, it is important that we only always test one of our parts, because otherwise this approach would fall apart as well. Therefore, in all our testing, we always only tested one part at a time. Now, after we got out the bottom lines, let's look into some of the results we have gathered. First, the 3' UTRs that we have characterized in protoplast transformation. On the left side, you can see our results in Taraxacum officinale, which is the dandelion plant that is just growing in the middle of Germany, basically everywhere. Where you can see also very varying expression strength with expression strength with these parts, ensuring that these parts can actually be used for our approach, because obviously if you want to work with the plant, you want to have access to different expression strength of your parts. And on the other side, you can see Taraxacum, the results in Taraxacum kok-saghyz, so the rubber-producing dandelion plants, with also varying expression strength, which again is nice to see. Now, these results are very exciting for us, but might not be as exciting if you are not working with dandelion in the future yourself. Therefore, we also made sure that we tested these genetic parts also in the model plant Nicotiana benthamiana. You can see on the left-hand side our promoter of 5' UTR constructs, which also needed some expression strength, also varying expression strength, showing these are functional in Nicotiana, and our 3' UTR constructs also being functional in the same plant. You might have noticed that these are quite a few constructs we have worked with, and this is also a possibility where I would like to use this opportunity to thank Twist, because they synthesized all of these 3' UTR all these 3' UTRs for us, because otherwise, in the relatively short scope of an iGEM season, this would not have been possible any other way. Now, another issue for you might be that we want to work with a dandelion plant that in the end works and grows in the field outside. Therefore, you need a stably transformed plant. And you might be wondering how we did this, because as I mentioned, an iGEM season is rather short, only being half a year. So stable plant transformation would prove challenging in most approaches. Therefore, we incorporated the cut budding protocol, which is a relatively novel protocol for dandelion published by Kao et al., where you take a fully grown dandelion plant, then you cut off small root segments, immerse it in agrobacterium solution, and then you just basically let it lay around for 2 weeks. And genetically modified or transgenic roots and shoots will grow from it, and then you have your stably transformed plant material. And we tested our parts, obviously, also with this method, where we can see, for example, here for the tubulin promoter 5' UTR, successful stable transformation in the roots of this dandelion, showing that at least some of our parts are functional in stably transformed dandelion plants, and this could also be investigated further in the future. Now with this, we have now all the parts together to form a well-adapted industrial crop, which in a nutshell means we have facilitated the genetic access to this crop, which makes it possible to adapt the plant traits, so seed size, root architecture, and leaf morphology, which in turn will lead to us using an additional rubber source and the reduction of environmental impact. Talking about environmental impact, of course, it is always important if you work in a project such as ours, that you don't just look on your project from a lab perspective, but also look outside the lab for the people that will actually be using your plants, because we will probably not be planting dandelion outside. Therefore, we talked to different stakeholders as well to get an evaluation in our project in that regard. For example, from a legal perspective. You might be aware that in Europe in general, and especially in Germany, it is very difficult to get a genetically modified plant out in the field. This is why we also took into the account the upcoming EU regulations on novel genomic techniques, the NGT regulations, which basically say if your genetically modified crop is indistinguishable by a normally bred plant, it will just be handled as a normally bred plant. This is why we had such an intense focus on using cisgenic parts, so genetic material that was originally also from the dandelion, because then we can just put it back into the dandelion, and after this novel NGT regulation, it will be considered a normally bred plant and therefore not fall under the genetically modified plant umbrella. Secondly, the economical perspective was important to us. This is why we also talked to stakeholders such as Continental, the big tire manufacturer, or also to farmers, which in the end need to plant our dandelion plants. because they told us that it is very expensive to buy new equipment to facilitate or to harvest new plants that don't fit their old equipment. This is also why we had this focus on changing leaf morphology, because then farmers can easier use all the equipment they already have, making it easier for dandelion to be grown here. And lastly, sustainability, of course, because in the end, we will bring a new plant into the German environment, which obviously always is associated with risks. This is why we also talk, for example, to the Julius Kühn Institute, which is the head in Germany, basically, for investigating invasive plants or novel plants into the German ecosphere. And what they told us is that dandelion in and of itself and with the modifications we plan is probably safe for the use in the German climate. Which brings us to the end, and I want to use this opportunity to thank all our sponsors that have supported us along the way. And a few special thanks to the University of Marburg, which lent us all the equipment, and also the Institute for Synthetic Microbiology, also based in Marburg, which helped us along a lot. And of course, a big shout out to all of our team for their dedication and all of the sleepless nights throughout the entire season last year. And Twist Bioscience for sponsoring us, of course, with these genetic parts I have mentioned earlier, and Bioscience Bio for having us today. And now, if you have any further questions, feel free to ask them, and we are happy to answer them. Thank you.
Thank you so much, Alexander and Julia. That was such an inspiring presentation. We do indeed have a few questions from the audience. If anyone else has a question, please feel free to post this in the questions box at the top right of your screen. So the first question I have for you is, why did you choose the dandelion plant and not other species with similar challenges or circumstances?
In the beginning, we originally wanted to go into not completely different project. We were interested in finding novel food sources, but then we stumbled more by coincidence over this dandelion plant, and its rubber producing capabilities was quite kind of interesting. And then we read more upon rubber production in general and saw that there's huge problems associated with it, with the labor exploitation, the massive rainforest deforestation that is going on and still going on and probably continuing for a long time. And we were like, this is a great opportunity, interest is there, and we also wanted to make it easier for other people to work with this plant, because in the end it wasn't possible previously as easily because the genetic access just wasn't there at all.
That makes sense. Thanks. And a question that came in is, could you please explain how ruby red helps in profiling the genetic expression? Because you showed that on one slide quite quickly, but
so the Ruby reporter is, maybe I can go to the slide back so you might be able to see, here. With coloring, because the Ruby reporter is are 3 enzymes that produce over a cascade this betalain pigment, which accumulates over time, which makes it just a very sensitive qualitative control when trying to test if a transformation has actually worked. Of course, there's always the issue with having a similar color to stressed plants, because they also might appear red, but this is a different pigment to the stress pigment that plants produce. And since it is really easy to accumulate, rather easy to work with, you don't have the issue that you have with fluorescence reporters, for example, because for fluorescence reporters, you need specialized equipment, for example, a fluorescence microscope or a plate reader or something. And with Ruby Red, you just transform it, and the leaf is either red or the leaf is not red. So it's very easy for quick screening.
Thank you. This question is quite long, so I'll start by reading the first half. Did you isolate the promoter from the dandelion plant itself and use that to construct a vector for driving your targeted gene editing?
I think I didn't understand the entire question, but so maybe I can rephrase it. Because so what we did, we took the entire promoter fragment UTR completely from the dandelion genome. So we amplified it using PCR, and then we cloned it into our measurement construct I showed earlier, this ratiometric approach, and then transformed it into dandelion using agrobacterium-mediated transformation or using protoplast transformation. So, but I don't know if this answers your question entirely.
I'm just reading the rest of the question. I think that answers some of the rest of the question, because the follow-up was, could you explain how you identified the promoter region, and then how did you validate promoter activity, and how did you use it to construct a vector?
Sure. So we identified a promoter so we weren't going into it and we were like, aha, this is the promoter. This wasn't possible in the scope of our project. What we did, we looked using this RNA sequencing data. This was publicly available, built our transcriptomic pipeline, and looked in the general genes that were very highly expressed and with low variance. And then the coding sequence is relatively easily to see in the genome, as you know. And then we just went and took 2 kilobases upstream of this coding sequence. Just everything of it, we used PCR, took it out, and then used Golden Gate cloning to clone it into our testing constructs.
Thanks. I'm going to jump ahead to a question that just came in because it's related to this as well. It's actually 2 questions, and I think you just answered the first one already because it's also about the promoter. What are your selection criteria for the promoter, and how did you deal with the long promoter sequence? But this question is interesting because it starts by saying, thank you so much for the talk. I'm from an iGEM team participating this year. I would like to ask some questions. So that was one question was about promoters as well. And the second question they're asking is, how do you measure rubber output from the transgenic dandelion plants?
So you mean isolating, when you isolate the rubber afterwards?
Yeah.
The thing is, we did not get to that point having that we get out or isolate actually the rubber out of these out of the plants, because the time was just too short, and item season is just half a year. Mm-hmm. But what you can do to measure is just extracting it out of the roots and then comparing it to the weight of the plant. So that would be one method to do that.
Thanks. And then this wasn't a question, but this is just some feedback. It says, I knew nothing about what you have been discussing, but it has been totally fascinating to learn about all these new biological innovations. So that was just a nice comment.
Thank you very much.
And we got a follow-up question from the promoter talk. Why 2K base pairs upstream?
Theoretically, of course, you can just go as long as you want. Obviously, at some point you will run into other regulatory elements and other genes. But 2 base pairs is how it was done in a similar approach when using Arabidopsis as an organism, when they tried to find promoters in there. And in general, it is regarded as highly likely if you take these 2 kilobases upstream of the coding sequences, is that the core promoter element and cis-regulatory elements will be inside this region. I mean, of course, plants are weird sometimes, and not all of our promoters will be complete, and not all of them were functional, for sure. Mm-hmm. So there will be issues, but for most of the promoters, it is highly likely that that most of the promoter is there and will be functional in plants.
Thanks. We got a lot of questions coming at once just now. Is root tip budding method of transformation as efficient or better than agrobacterium-mediated in vitro regeneration? How did you regenerate the transformed putative plants?
So we don't know how the regeneration would be with other methods for stable transformation, because we just didn't do it. But we had very high transformation efficiencies, especially for transformed root material. It was very successful for dandelion, if I recall correctly, but I might be off here. I think we had transformation efficiencies with 30%, partly with it. If you are more interested in how well it went, I can recommend you to our wiki, which is our website where all of our results is. So iGEM Marburg wiki, 2024. So everything should be there. But the nice thing for cutted budding is really the high throughput you can have with it, because it's very easy to do and really fast. And which is also important to mention, cutted budding is still agrobacterium mediated, because you will dip the root segment in agrobacterium solution. So it doesn't work if you don't use agrobacterium.
Mm-hmm. Thanks. And this question says, what are the genes that you expect to express in order to increase rubber production?
I mean, we did not look too closely into that, since it was not one of our aims. As we said earlier, it's more about other agricultural traits. The rubber yield is actually not the problem that is hindering the dandelion from being cultivated on a large scale. So that's why we did not look that much closer into that, I think.
We didn't really look into rubber, higher rubber production, because the current plants are at their limits as far as we know, and the bottleneck is with other agricultural traits.
Thanks. And the next question is a question that I also had, actually. Do you guys have any plans for the future of this project, like making a startup or continuing the research or anything like that?
Not a startup is currently not planned, but for sure this is something we don't want the story to end. We don't want the potential of dandelion to get lost, because it has so many potentials, not just for rubber production, but also things we looked also into bioremediation, or as a biofuel, probable applications in medicine, things that you can think of, that we just did not have the time to look really closely into that. There are people from our team who are currently still working on the toolbox, who are currently doing their, I think, bachelor thesis on. So there are plans and work is being done that this work continues, but we for sure hope that also other researchers, maybe also other iGEM teams, continue with the work that we have built, and that, the potential can be used.
Thanks. This next question says, thank you for this presentation. It is awesome, and I am curious about the sustainable environment activity. Could you give us a framework to do this activity on legal, economic, sustainability?
Can you repeat the question? I didn't quite get it.
Could you give us some framework for this activity? So for sustainable environmental activity, I guess some context on the legal, economical, and sustainability of it.
So at first you need to think about who your project will impact, or who you want to impact with your project, because in the end, our dandelion plants shall produce rubber, but then we need to ask who will plant the dandelion plant for us, and in this case it was for the farmers. Then you run into issues. Okay, what if you have, I don't know, 100 hectares or 1,000 hectares suddenly full of Russian dandelion? Might this be a problem for the ecosystem if there are, I mean, you have seen dandelion and you have seen these nice parachutes with seeds flying everywhere. Might this be an issue if there are suddenly a bazillion seeds of dandelion flying around? And so this was also a concern, and this is why we talked then to biosafety experts in this regards. So it depends on the project you're working with. It's really important just to see what do you want your project to achieve. It might not be needed for your project, depending on what you do, if to talk to an economical perspective, from an economics perspective, because if you are just developing a technique for other people in the lab to use it, then you just need to talk with other researchers what makes it easier for them to use it in the lab. But if you want your project to be mass produced, for example, as ours, then you need to also get in touch with people that have the resources to do it.
Thanks. I've got a few questions left. One is, what was the biggest challenge that you both encountered in this project?
I definitely think the time, the restriction of time, but and then we had so many ideas that we wanted to do, so many things in the lab that we wanted to try, so many people we wanted to talk to, and it was just, yeah, I think prioritizing on what we can actually do in that limited amount of time. I think that was the most challenging thing for us, I think. Or do you have any?
No, I definitely agree that the time scale in working with plants for half a year is rather short. So this was the main issue.
Thanks. And there are a few questions that are quite specific to very specific projects. So rather than reading these out loud, I'm just going to make sure that you get them and you can answer them individually, because then we don't all have to listen to all the specific technical details. And with that, I'll just have one question left. Do you think that this project that you have developed could be considered a proof of concept that could also be applicable in other environments? Or is it very specific to dandelions and rubber?
I think so. I mean, natural rubber isn't the only issue that we have, a resource, a problem that we can solve with synthetic biology in general. And I think since we have so many environmental challenges in general in the world, I mean, it's not just the natural rubber crisis or problems that we have there. I think looking for alternatives and other or diversifying natural resources is in general an idea that we, in my opinion, should follow. And yeah, maybe there are other dandelions that we can look for.
Yeah. Well, thank you so much. That brings us to the end of this webinar. So thank you again, Julia and Alexander, for a very interesting presentation and a great discussion. And thanks, of course, to our sponsor, Twist Bioscience. And finally, thanks to the audience for taking the time to attend and listen in today. So until next time, good luck in your research, and goodbye from all of us at Twist Bioscience and Bite Size Bio.
Goodbye.
Bye.